Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 6.5, regulation of gene expression. So in 6.3 and 6.4 we discussed the process of gene expression, and today we'll be discussing how it's regulated, looking at epigenetic changes, factors that affect transcription, factors that affect translation, and also differences in prokaryotes and eukaryotes in regulating gene expression. And our suggested skill is argumentation 6a, make a scientific claim. So every cell in an organism has the exact same genome, but not every gene is expressed in every single cell. Different cells have different roles, they have different proteins they need to produce, so this expression is regulated. And this can happen both before and after transcription and before and after translation. And we start by talking about an epigenetic change, which is a change where the DNA sequence is not changed, but its expression is still affected. And so this happens through DNA packing, where the more tightly bound the DNA is around histone proteins, the less accessible it is for transcription. So DNA methylation makes the DNA tighter, more tightly bound around histone proteins, while histone acetylation makes it looser. Genes can be constitutive or inducible, meaning they're either always expressed or they need to be activated in order to be expressed. And so regulatory sequences are stretches of DNA that interact with regulatory proteins in order to control this gene expression. And this gene expression can either be promoted through an inducer or blocked through a repressor. And so when we talk about starting gene expression through transcription, we look at the transcription initiation complex, where an activator protein has to bind to an enhancer and bind to general transcription factors so that DNA can bend, bringing this complex to the promoter, and only then can the promoter be activated and RNA polymerase bind to it. So these transcription factors have to be present, and this enhancer has to function in order for transcription to begin. And so this can also be coordinated, so multiple genes can be activated at the same time by certain transcription factors, helping regulate gene expression. Regulation also occurs post-transcription. And so we discussed some of this when talking about mRNA processing in 6.3, but when we do have RNA that is being processed after transcription, some non-coding regions are cut out of it. This is known as RNA splicing, and the non-coding regions are known as introns. And so we have alternative RNA splicing, where the same mRNA strand can have different coding and non-coding regions. So for one situation, the introns and the exons could be different, so depending on the goal of gene expression. So the parts that get cut out of the code of the genetic information are going to be different for different types of gene expression. So even the same mRNA strand can code for different proteins. And now additions to mRNA include the 5' cap and the poly-A tail, which help carry this message over to the ribosomes. And the longer poly-A tail allows mRNA to survive longer, and by protecting it from exonucleases, which and exonucleases are enzymes that are trying to degrade and obtain free nucleotides, and so occasionally they will cut off parts of the mRNA strand, but the longer the poly-A tail, the more non-coding nucleotides exist on that mRNA strand to protect the actual coding regions from these exonucleases. And it's important to remember that the methods of regulation we've been discussing so far have been eukaryotic, because processing of mRNA does not occur within prokaryotes. And now this is also going to be eukaryotic. We have translation and post-translation regulation. So again, regulatory proteins can be needed to activate or block translation, and after translation occurs, protein activation itself can be inhibited, because in order for a protein to be functional, it needs to fold. It needs to have this primary, secondary, tertiary, and quaternary structure, and if it doesn't achieve that, then the protein cannot be activated. Additionally, after translation, when the protein is tagged with ubiquitin, that signals to the cell that it needs to be destroyed, and so the protein will be destroyed by what's known as a proteasome. So regulation can happen even after translation, even after the protein has been created, before the protein can be functional and active within the cell. Now we need to discuss regulation within prokaryotes, and this happens through what are known as operons. And in bacteria, genes are clustered into groups, and these groups are known as operons, and they're controlled as one unit. And so operons consist of a few different parts. There's going to be the genes, there's going to be the operator, which controls access of RNA polymerase to the gene. And of course, RNA polymerase needs to be able to access the genes in order to create transcription. And if the operator is bound by a repressor, then expression does not work. RNA polymerase cannot access the genes. And the promoter is where RNA polymerase attaches, just like in eukaryotes. So if we look over here at this operon, we see our promoter, that's where RNA polymerase is attaching. But when there's a repressor bound to the operator, then the polymerase cannot get to the genes and result in transcription. And this is similar to transcription factors inducing transcription for multiple genes. So this can also be coordinated because these genes are clustered into operon groups. And so there are two types of operons. There's an inducible operon that is normally off, and it needs to be turned on in order for the transcription to happen. So the operon is normally off, meaning the repressor is normally on, because that means transcription cannot happen. And generally, inducible operons are catabolic, because you're only breaking down molecules when you need to. And so one of the common inducible operons is the lac operon for lactose digestion. So lactose digestion enzymes are only produced when needed. So when the correct inducer, in this case allolactase, binds to the repressor protein, then it will inactivate the repressor protein, allowing the operon to turn on. So without lactose, the operon is not working. RNA polymerase cannot transcribe the genes to create lactose digestion enzymes because they're not needed in the cell. However, when lactose enters the cell and we have allolactose within the cell, that's going to bind to the repressor, causing it to leave the operator, and now RNA polymerase is able to transcribe these genes in order to synthesize these lactose digestion enzymes in order to get rid of the enzymes within the cell, in order to get rid of the lactose within the cell. Now, the other type of operon is a repressible operon, and this is an example of this is a tryptophan operon. And this is normally on, so the repressor is off, and it's anabolic, it's for building. And you generally want to be building these and putting together these molecules until you have too many of them. And so when there is too much tryptophan in the cell, it binds to the repressor, activating the repressor and turning the operon off. So a repressible operon is innately on and the repressor is off until there's too much of the product. And that's sort of like a feedback regulation of gene expression. And so connecting gene expression to phenotype, which again is your outward appearance, the phenotype of a cell or an organism is determined by the combination of genes that are expressed and at the levels at which they are expressed. So the function of the proteins that are resulting from this gene expression and also the amount of each protein. And transcription begins during development, and from there we have sequential gene expression going on from each cell, and so cell differentiation results from the expression of genes for tissue-specific proteins. So all of your cells are differentiated within the eukaryotic organism. They all have a different task, and they all start from stem cells, but then the regulation of gene expression is going to result in different proteins being produced, and so we get different kinds of cells: intestinal cells, liver cells, cardiac cells, nerve cells, blood cells, and muscle cells that are all going to be coming from one base cell that has the same genome, but the regulation of these genes results in a different phenotype. So lots of different kinds of expression here. In prokaryotes, we looked at operons, which can both be inducible and repressive. We looked at eukaryotic regulation through gene packing, transcription factors, post-transcription processing, translation, protein tagging. And so there's so many different ways that gene expression is regulated. And then it's also crucial to note the connection between phenotype and gene expression, because all of the cells in our body have the same genome. However, they all have a different phenotype based on their function, which is a result of gene regulation and which genes are expressed.